Chapter 3, Cells. Recall in Chapter 1 that we suggested that the body may be considered a city of cells and a container of chemicals. Chemicals are non-living. Cells are living. What is that can change a particular assortment of non-living chemicals into a living cell capable of reproducing itself? We don't know. We do know that such a transformation occurred at least once several billion years ago, and these billions of years later, here we are, each of us a fine city with a population of trillions of cells. Cells are the smallest living subunits of a multicellular organism, such as a human being. A cell is a complex and precise arrangement of the organic and inorganic chemicals discussed in the previous chapter, and it carries out specific activities. Microorganisms such as amoebas and bacteria are single cells that function independently. Human cells, however, must work together and function interdependently. Homeostasis depends on the contributions of all the different kinds of cells. Human cells vary in size, shape, and function. Most human cells are so small they can be seen only with the aid of a microscope and are measured in units of micrometers. A meter, micrometers, formerly called microns. 1 micrometer equals one million of a meter, or 1/25,000 of an inch. One exception is the human ovum or egg cell, which is about 1 millimeter in diameter. Just visible to the unaided eye, some nerve cells, although microscopic in diameter, may be quite long. These in our arms and legs, for example, are at least 2 feet long. With respect to shape, human cells vary greatly. Some are round or spherical, others are rectangular, and still others are irregular. White blood cells even change shape as they move. Cell functions also vary, and because our cells do not act independently, we will cover specialized cell functions as part of tissue functions in Chapter 4. On the basis of function, there are more than 200 different kinds of human cells. This chapter is concerned with the basic structure of cells and the cellular activity common to all or most of our cells. Despite their many differences, human cells have several similar structural features. A cell membrane, a nucleus and cytoplasm, and cell organelles. Red blood cells are an exception because they have no nuclei when mature. The cell membrane forms the outer boundary of the cell and surrounds the cytoplasm, organelles, and nucleus. Cell membrane, also called the plasma membrane. The cell membrane is the outer boundary of a cell and is made of phospholipids, cholesterol, and proteins. The arrangement of these organic molecules is shown in Figure 3.1. The phospholipids are diglycerides and they form a bilayer or double layer which makes up most of the membrane. Phospholipids permit lipid-soluble material to easily enter or leave the cell by diffusion through the cell membrane. The presence of cholesterol decreases the fluidity of the membrane, thus making it more stable. The proteins have several functions. Some form channels to permit passage of materials such as water or ions. Others are carrier enzymes or transporters that also help substances enter the cell. Still, other proteins with oligosaccharides on the other surface are antigens, markers that identify the cell of an individual as self. These self-antigens are used by the white blood cells of the immune system to distinguish pathogens, in parentheses, foreign antigens, close parentheses, from the cells that belong in the body. Other membrane proteins form intercellular junctions, attachment points with neighboring cells. This contributes to the strength and resistance of tearing of some tissues. Yet another group of proteins serve as receptor sites for hormones, growth factors, and other signaling chemicals. These chemicals bring about their specific effects by first bonding to a particular receptor on the cell membrane, a receptor with the proper shape. This bonding or fit then triggers chemical reactions within the cell membrane or the interior of the cell. See box 10.3 in chapter 10 for an illustration involving the hormone insulin. The cell membrane of a single human cell has hundreds of different receptors. All of these molecules that bond to the cell membranes are part of the chemical communication network of our cell, a part of the chemical communication networks our cells have. An unavoidable consequence of having so many receptors for chemical communication is that some pathogens have evolved shapes to match certain receptors. For example, HIV, the virus that causes AIDS, just happens to fit a particular surface receptor on our white blood cells. When the virus fits in, the receptor becomes a gateway into the cell, which begins the takeover of the cell by the virus. Most often, however, the cell membrane is a beneficial structure. Although the cell membrane is the outer boundary of the cell, it should already be apparent to you that it is not a static or wall-like boundary, but rather an active, dynamic one. It is much more like a line of toll booths than a concrete barrier. The cell membrane is selectively permeable, that is, certain substances are permitted to pass through and others are not. These mechanisms of cellular transport will be covered later in this chapter. The cell membrane is of particular importance for muscle cells and nerve cells because it carries electrical impulses. This topic will be covered in chapter 7 and 8. Nucleus. With the exception of mature red blood cells, all human cells have a nucleus. The nucleus is within the cytoplasm and is bounded by a double-layer nuclear membrane with many pores. It contains one or more nuclei and the chromosomes of the cell. A nucleus is a small sphere made of DNA, RNA, and protein. The nuclei form a type of RNA called ribosomal RNA, which is assembled with protein to become ribosomes, in parentheses, a cell organelle, close parentheses, and is involved in protein synthesis. The nucleus is the control center of the cell because it contains the chromosomes. The chromosome number of humans is 46, and these are in 23 pairs, maternal and paternal. Pairs 1 through 22 are called autosomes. The remaining pair consists of the sex chromosomes, designated XXX for women and XY for men. Even with a microscope, the chromosomes of a human cell are usually not visible. They are long threads called chromatin. It is estimated that the DNA of a cell's 46 chromosomes, if uncoiled and placed end to end, would be about 6 foot long. Before a cell divides, however, the chromatin coils extensively into visible chromosomes. Chromosomes are made of DNA and protein. Some chromosomal proteins provide the structural framework for the coiling of the chromatin into chromosomes so that the cell division can take place. Other chromosomal proteins are enzymes that help regulate the activity of the DNA. Remember from our earlier discussion that the DNA is the genetic code for the characteristics and activities of the cell. Although the DNA in the nucleus of each cell contains all of the genetic information for all human traits, only a small number of genes, in parentheses, a gene is a genetic code for one protein, close parentheses, are actually active or switched on in a particular cell. These active genes are the codes for the proteins necessary for the specific cell type and its functions. For example, the gene for insulin is present in all human cells, but only in certain islets cells of the pancreas is the gene active or switched on. Only in these cells is insulin produced. Similarly, the genes for antibody production are present in all human cells, but only in certain lymphocytes are these genes active. Only these lymphocytes produce antibodies. How the genetic code in chromosomes is translated into proteins will be covered later in this section. Cytoplasm and cell organelles. Cytoplasm is a watery solution of minerals, gases, organic molecules, and cell organelles that is found between the cell membrane and the nucleus. Cytosol is the water portion of the cytoplasm, and many chemical reactions take place within it. Cell organelles, from the Latin word little tools, are intracellular structures often bounded by their own membranes that have specific functions in cellular metabolism. The endoplasmic reticulum, ER, is an extensive network of membraneous tubules that extend from the nuclear membrane to the cell membrane. Rough ER has numerous ribosomes on its surface, whereas smooth ER has no ribosomes. At a network of interconnected tunnels, the ER is a passageway for the transport of the materials necessary for cell function within the cell. These include proteins synthesized by the ribosomes on the rough ER and lipids synthesized by the smooth ER. Ribosomes are very small structures made of protein and ribosomal RNA. Some are found on the surface of rough ER, whereas others float freely within the cytoplasm. Ribosomes are the site of photosynthesis. The proteins produced may be structural proteins such as collagen in the skin, enzymes, or hormones such as an insulin that regulates cellular processes. These proteins may function within the cell or be secreted from the cell to be used elsewhere in the body. Our protein molecules are subject to damage, and some cellular proteins, especially regulatory proteins, may be needed just for a very short time. All such proteins must be destroyed, and this is the function of proteasome. Proteasomes. A proteasome is a barrel-shaped organelle made of enzymes that cut protein molecules apart. Protease enzymes. Proteins that are to be destroyed, that is, those no longer needed or those that are damaged or misfolded, are tagged by a protein called ubiquitin, in parentheses, sort of a cellular mop or broom, close parentheses, and carried into a proteasome. The protein is snipped into peptides or amino acids, which may be used again for protein synthesis on ribosomes. Proteasomes are particularly important during cell division and during embryonic development, when great changes are taking place very rapidly as cells become specialized. Many of our cells have secretory functions. That is, they produce specific products to be used elsewhere in tissues. Secretion is one of tasks of the Golgi apparatus, a series of flat membranous sacs, somewhat like a stack of saucers. Carbohydrates are synthesized within the Golgi apparatus and are packed along with other materials for secretion from the cell. Proteins from the ribosomes or lipids from the smooth endoplasmic reticulum may also be secreted in this way. To secrete a substance, small sacs of the Golgi membrane break off and fuse with the cell membrane, releasing the substance to the exterior of the cell. This is exocytosis, exo meaning to go out of the cell. Mitochondria are oval or spherical organelles bounded by a double membrane. The inner membrane has folds called cristae. Within the mitochondria, the aerobic, oxygen-requiring reactions of cell respiration take place. Therefore, mitochondria are the site of ATP, in parentheses, and hence energy, close parentheses, production. Cells that require large amounts of ATP, such as muscle cells, have many mitochondria to meet their need for energy. Athletic training increases the number of mitochondria within a muscle cell. Brown adipose tissue has abundant mitochondria because it is a heat-producing tissue. Mitochondria contain their own genes, 37 of them in a single circular DNA molecule. They duplicate themselves within a cell when a cell divides. An individual's mitochondrial DNA, mtDNA, is usually of maternal origin. That is, from the mitochondria that were present in the ovum or egg cell, which was then fertilized by a sperm cell. The mitochondria of the sperm cell usually do not enter the ovum during fertilization because they are not found in the head of the sperm with the chromosomes. The 37 genes of mitochondrial DNA code for only 13 proteins, all of which are involved in energy production. More than 400 different mutations or errors may occur in these genes, and the disorders that result are invariably severe and often fatal. Women who are found to have such mtDNA mutations may choose to use a form of in vitro fertilization involving the cytoplasm of donated egg cells. Lysosomes are single membrane structures that contain digestive enzymes. When certain white blood cells engulf bacteria, the bacteria are digested and destroyed by these lysosomal enzymes. Worn out cell parts, old proteins, and dead cells are also digested by these enzymes. The products, simple chemicals, may then be recycled. This is autophagy, self-eating, a beneficial process that is necessary to keep cells healthy. Autophagy also cleans up damaged parts after injury so that tissue repair can begin, but it does have a disadvantage in that lysosomal digestion contributes to inflammation in damaged tissues. And excess of inflammation can start a vicious cycle, actually a positive feedback mechanism that results in even more extensive tissue damage. The opposite, impaired autophagy, can result in the accumulation of damaged proteins or cell parts within cells. Some degenerative diseases of the nervous system and cardiovascular system are thought to be the result of lysosomal malfunctioning, as are some autoimmune diseases and the aging process. Many of our cells are capable of dividing or reproducing themselves. Centrioles are a pair of rod-shaped structures perpendicular to one another, located just outside the nucleus. Their function is to organize the spindle fibers during cell division. The spindle fibers are contracting proteins that pull the two sets of chromosomes apart toward the ends of the original cell as it divides into two new cells. Each new cell then has a full set of chromosomes. Flagella and cilia are thread-like projections through the cell membrane. Each is anchored by a basal body just within the membrane. Flagella are usually long, cilia are shorter. Many species of bacteria are protozoa. Many species of bacteria and protozoa have one or several flagella to enable them to move. The only human cell with a flagellum is the sperm cell, and the flagellum provides the motility or movement. Human cells may have two kinds of cilia. Motile cilia occur in bunches or swaths on the free surface of one type of epithelial cell. They resemble small fields of wheat. These cilia beat or bend in unison and sweep materials across the cell surface. Cells lining the trachea, for example, have cilia to sweep mucus and trap dust up toward the pharynx. In women, cells lining the fallopian tubes have cilia to sweep the egg cell toward the uterus. Most human cells have a sensory cilium called a primary cilium. The single cilium is part of a cell's communication system. and has been likened to an antenna. Just as the antenna of a radio can pick up signals from hundreds of stations, a primary cilium can detect many different kinds of changes. Some of these changes are chemicals produced by other cells. Other changes are mechanical, such as the flow of blood or tissue fluid in the cell's environment. Primary cilia seem to be essential to proper mitosis and development of an embryo, in parentheses, including the left-right asymmetry of thoracic and abdominal organs, close parentheses, and for many functions in established tissues as well. For example, the rods and cones of the retina of the eye, the receptors for light are primary cilia. Again, for example, the rods and cones of the retina of the eye, the receptors for light, are primary cilia. Microvilli are folds of the cell membrane on the free surface of a cell. These folds greatly increase the surface area of the membrane and are part of the cell's lining organs that absorb materials. The small intestine, for example, requires a large surface area for the absorption of nutrients, and many of its lining have microvilli. Some cells of the kidney tubules also have microvilli that provide for efficient reabsorption of useful materials back to the blood. The cytoskeleton is just what its name suggests, the framework or support of a cell. Microfilament made of a protein actin help support the cell membrane, and microvilli, if they are present, give shape to the cell and contribute to the movement of the cell. When you think of movement, you may rightly think of muscle cells, as you will see in chapter 7. Actin is one of a muscle cells contracting proteins. Other protein filaments provide for the support of organelles within the cell and help form junction or linkage sites between adjacent cells. The functions of the cell organelles are summarized in Table 3.1. Takeaways. 1. Name the 3 types of organic chemicals that make up cell membranes. 2. What is the human chromosome number? What are chromosomes made of? Which of these is the genetic code? 3. Name the cell organelles that are the site of the following: ATP production, protein synthesis, lipid synthesis. Takeaways. 1. Which cellular transport mechanisms may be considered passive, in parentheses, as opposed to active, close parentheses, and why? 2. Which cellular transport mechanism depends on lysosomes for its completion? 3. Which cellular transport mechanism can change a hypertonic solution to an isotonic solution? Cellular transport mechanisms. Living cells constantly interact with the blood or tissue fluid around them, taking in some substances and secreting or excreting others. Several mechanisms of transport enable cells to move materials into or out of the cell. Diffusion, osmosis, facilitated diffusion, active transport, filtration, phagocytosis, and pinocytosis. Some of these take place without the expenditure of energy by the cells. Others, however, do require energy, often in the form of ATP. Each of these mechanisms is described in the following sections, and an example is included to show how each is important to the body. Diffusion is the movement of molecules from an area of greater concentration to an area of lesser concentration, in parentheses, that is, with or along a concentration gradient, close parentheses. Diffusion occurs because molecules have free energy, that is, they are always in motion. The molecules in a solid move very slowly. Those in a liquid move faster, and those in a gas move faster still, such as when ice absorbs heat energy, melts, and then evaporators. Imagine at the bottom of a glass of water or a green sugar cube, green so that we can see it. As the sugar dissolves, the sugar molecules collide with one another, or the water molecules, and the green color seems to rise in the glass. These collisions spread out the sugar molecules until they are evenly dispersed among the water molecules. This would take a very long time, and the water eventually becomes entirely green. The molecules are still moving, but as some go to the top, others go to the bottom, and so on. Thus, an equilibrium, in parentheses, or steady state, close parentheses, is reached. Diffusion is a very slow process, but it can be an effective transport mechanism across microscopic distances. Within the body, the gases oxygen and carbon dioxide move by diffusion. In the lungs, for example, there is a high concentration of oxygen in the alveoli air sacs and a low concentration of oxygen in the blood in the surrounding pulmonary capillaries. The opposite is true for carbon dioxide. A low concentration is in the air in the alveoli, and a high concentration is in the blood and the pulmonary capillaries. These gases diffuse in opposite directions, each moving from where there is more to where there is less. Oxygen diffuses from the air to the blood to be circulated throughout the body. Carbon dioxide diffuses from the blood to the air to be exhaled. Osmosis. Osmosis may be defined simply as the diffusion of water through a selectively permeable membrane. That is, water will move from an area with more water to an area with less water. Another way to say this is that water will naturally tend to move to an area where there is more dissolved material, such as salt or sugar. If a 2% salt solution and a 6% salt solution are separated by a membrane, allowing water but not salt to pass through it, water will diffuse from the 2% salt solution to the 6% salt solution. The result is that the 2% solution will have become more concentrated and the 6% solution will become more diluted until the concentrations reach equilibrium. In the body, the cells lining the small intestine absorb water from digested food by osmosis. These cells have first absorbed salts and have become more salty, and water follows salt into the cells. The process of osmosis also takes place in the kidneys, which reabsorb large amounts of water, many gallons each day, to prevent its loss in urine. Terminology of solution lists some terminology. Box 3.1, terminology of solution, lists some terminology we use when discussing solutions and the effects of various solutions on cells. Facilitated diffusion. The word facilitate means to help or assist. In facilitated diffusion, molecules move through a membrane from an area of greater concentration to an area of lesser concentration, but they need some help to do this. In the body, our cells must take in glucose to use for ATP production. Glucose, however, will not diffuse through most cell membranes by itself, even if there is more outside the cell than inside. Diffusion of glucose into most cells require a glucose transporter, which may also be called a carrier enzyme. These transporters are proteins that are part of the cell membrane. Glucose bonds to the transporter and by doing so changes the shape of the protein. This physical change propels the glucose into the interior of the cell. Other transporters are specific for organic molecules such as amino acids. Active transport. Active transport requires the energy of ATP to move molecules from an area of lesser concentration to an area of greater concentration. Notice that this is the opposite of diffusion, in which the free energy of molecules causes them to move to where there are fewer of them. Active transport is therefore said to be movement against a concentration gradient. In the body, nerve cells and muscle cells have sodium pumps to move sodium ions, Na+, out of the cells. Sodium Ions are more abundant outside the cells and through specific diffusion channels. They constantly diffuse into the cell, their area or lesser concentration. Without the sodium pumps to return them outside, the incoming sodium ions will bring about an unwanted nerve impulse or muscle contraction. Nerve and muscle cells constantly produce ATP to keep their sodium pumps and similar potassium pumps working and prevent spontaneous impulses. Another example of active transport is the absorption of glucose and amino acids by the cells lining the small intestines. The cells use ATP to absorb these nutrients from digested food even when their intracellular concentration becomes greater than their extracellular concentration. Filtration. The process of filtration also requires energy, but the energy needed does not come directly from ATP. It is the energy of mechanical pressure. Filtration means that water and dissolved materials are forced through a membrane from an area of higher pressure to an area of lower pressure. In the body, blood pressure is created by the pumping of the heart. Filtration occurs when blood flows through capillaries whose walls are only one cell thick and very permeable. The blood pressure in capillaries is higher than the pressure of the surrounding tissue fluid. In capillaries throughout the body, blood pressure forces plasma, water, and dissolves material through the capillary membranes into the surrounding tissue spaces. This creates more tissue fluid and is how cells receive glucose, amino acids, and other nutrients. Blood pressure in the capillaries of the kidneys also bring about filtration, which is the first step in the formation of urine. Box 3.1 Terminology of solutions. Human cells or other body fluids contain many dissolved substances called solutes, such as salts, sugar, acids, and bases. The concentration of solutes in a fluid creates the osmotic pressure of the solution, which in turn determines the movement of water through membranes. As an example, here we will use sodium chloride, NaCl. Human cells have an NaCl concentration of 0.9%. With human cells as a reference point, the relative NaCl concentrations of other solutions may be described with the following terms. Isotonic, a solution with a salt concentration equal to that in cells. The blood plasma is isotonic to red blood cells. Hypotonic, a solution with a salt concentration lower than that in the cells. Distilled water, 0% salt, is hypotonic to human cells. Hypertonic, a solution with a salt concentration higher than that in the cells. Seawater, 3% salt, is hypertonic to human cells. Refer now to the diagram in box figure 3.1 of red blood cells, RBCs, in each of these different types of solutions and note the effect of each on osmosis. When RBCs are in plasma, water moves into and out of them at equal rates, and the cells remain normal in size and water content. If RBCs are placed in distilled water, more water will enter the cells than leave, and the cells will swell and eventually burst. If RBCs are placed in seawater, more water will leave the cells than enter, and the cells will shrivel and die. This knowledge of osmotic pressure is used when replacement fluids are needed for a patient who has become dehydrated. Isotonic solutions are usually used. Normal saline and Ringer's solution are examples. These will provide rehydration without causing osmotic damage to cells or extensive shifts of fluid between the blood and tissues. Phagocytosis and pinocytosis. These two processes are similar in that both involve a cell engulfing something, and both are forms of endocytosis. Endo meaning to take in, a cell. An example of phagocytosis is a white blood cell engulfing bacteria. The white blood cell flows around the bacterium, taking it in and eventually digesting it. Digestion is accomplished by the enzymes in the cell's lysosomes. Other cells that are stationary may take in small molecules that become absorbed or attached to their membranes. The cells of the kidney tubules reabsorb small proteins by pinocytosis so that the protein is not lost in urine. Table 3.2 is a summary of cellular transport mechanisms. Takeaways. 1. Which cellular transport mechanisms may be considered passive, in parentheses, as opposed to active, close parentheses, and why? 2. Which cellular transport mechanism depends on lysosomes for its completion? 3. Which cellular transport mechanism can change a hypertonic solution to an isotonic solution? The genetic code and the structure of DNA, RNA, and protein was described in Chapter 2. We will renew some of the essentials here and go a step further with a simple description of how all of these organic molecules are involved in the process of protein synthesis. DNA and the genetic code. DNA consists of two strands of nucleotides in the form of a double helix, very much like a spiral ladder. The uprights of the ladder are made of alternating phosphate groups and deoxyribose sugar molecules. The rungs of the ladder are made of the four nitrogenous bases, always found in complementary pairs, adenine and thymine, AT, and guanine with cytosine, G-C, or G-C, and A-T. Although DNA contains just these four bases, the bases may be arranged in many different sequences, reading up or down the ladder. It is the sequence of bases, the sequence of A, T, C, and G, that is the genetic code. The DNA of our 46 chromosomes may also be called our genome, which is the term for the total genetic information in the particular species. The human genome is thought to contain about 3.2 billion base pairs, and the number of our genes is estimated to be about 22,000. Recall from Chapter 2 that a gene is the genetic code for one protein. This is a simplification, and the functioning of gene is often much more complex. We have genes with segments that may be shuffled or associated in many combinations, or that function as networks, with the potential for coding for many more proteins. A full explanation is beyond the scope of our book, so for the sake of simplicity and in the following discussion, we will say that gene is the DNA code for one protein. Recall too that a protein is a specific sequence of amino acids. Therefore, a gene or segment of DNA is the code for the sequence of amino acids in a particular protein. The code for a single amino acid consists of three bases in the DNA molecule. This triplet of bases may be called a codon. A codon. There is a triplet of bases in the DNA for each amino acid in the protein. If a protein consists of 100 amino acids, the gene for that protein will consist of 100 triplets or 300 bases. Some of the triplets will be the same because the same amino acid may be present in several places within the protein. Also, part of the gene are other triplets that start and stop the process of making the protein. Rather like the way that capital letters or punctuation marks start and stop sentences, genes may also be influenced by chemical activity that has come to be called epigenetics, and we will consider this briefly before discussing the synthesis of proteins. Epigenetics. The prefix epi means upon, outer, or on top of, or in addition to. Epigenetics therefore means something in addition to genetics. The DNA and chromosomes, this sometimes may be changes. This sometimes may be changes in cellular structure or chemistry, or changes in the chromosomes themselves when certain genes are switched on and off. These are not mutations. The DNA genes remain unchanged, but the activity of the genes is altered. Gene expression is affected. The expression of a gene means that the product of the gene is somehow apparent to us in a way we can see or measure, or is not apparent when it should be. Examples would be having brown eyes or blue eyes, or producing or not producing the intestinal enzyme lactase to digest milk. Gene expression can be altered in several ways. Certain types of RNA molecules can suppress the activity of DNA genes. Parts of the chromosomes that are not codes for proteins, called non-coding DNA, also act as switches for the genes for proteins. This non-coding DNA was once called junk DNA before its function was discovered. Gene expression can also be affected by the attachment of simple chemicals to a chromosome. For example, methyl groups, CH3, are switches that inactive genes, and acetyl groups, C2H5, are switches that activate genes. Recent research suggests that environmental factors may influence these modifications of gene activity. Factors such as smoking or obesity are thought to affect gene expression in harmful ways, permitting lipid buildup in arteries, for example, whereas a factor such as regular exercise will alter gene expression in a beneficial way, preventing such buildup. We have so much more to learn about our genes. Well, we have so much more to learn about how diet, exercise, and even the air we breathe can affect our genes. But let us return now to the synthesis of proteins. RNA and protein synthesis. RNA has been found to have many functions. It contributes to the repair of DNA, and it is involved in gene expression. Although these functions of RNA are essential for us, the details are beyond the scope of our book. So the roles of RNA in the process of protein synthesis will be our focus. The transcription and translation of the genetic code in DNA into proteins requires several types of RNA. DNA is found in the chromosomes in the nucleus of the cell, but protein synthesis takes place on the ribosomes in the cytoplasm. Messenger RNA, mRNA, is the intermediary molecule between these two sites. When protein is made, or when protein is to be made, the segment of DNA that is its gene uncoils, and the hydrogen bonds between the base pairs break. Within the nucleus are RNA nucleotides and enzymes to construct a single strand of nucleotides that is a complementary copy of half the DNA gene. This process is transcription, or copying, and the copy of the gene is mRNA, which now has the condoms or the codons for the amino acids of the protein and will then separate from the DNA. The gene coils back into the double helix, and the mRNA enters leaves the nucleus, enters the cytoplasm, and become attached to the ribosomes. As a copy of the gene, mRNA is a series of triplets of bases. Each triplet is a codon, the code for one amino acid. Another type of RNA called transfer RNA, or tRNA, is also found in the cytoplasm. Each tRNA molecule has an anticodon, a triplet complementary to a triplet on the mRNA. The tRNA molecules pick up specific amino acids, in parentheses, which have come from protein in our food, close parentheses, and bring them to their proper triplets on the mRNA. This process is translation. That is, it is as if we were translating from one language to another, the language of nucleotide bases to that of amino acids. The ribosomes contain structural proteins and ribosomal RNA. rRNA that functions as an enzyme. Again, the ribosomes contain structural proteins and ribosomal RNA, in parentheses, rRNA, close parentheses, that functions as an enzyme called a ribosome to catalyze the information of peptide bonds between the amino acids. When an amino acid has been brought to each triplet on the mRNA and all peptide bonds have been formed, the protein is finished. The protein then leaves the ribosomes and may be transported by the endoplasmic reticulum to wherever it is needed in the cell, or it may be packaged by the Golgi apparatus for secretion from the cell. A summary of the process of protein synthesis is found in Table 3.3. Thus, the expression of the genetic code may be described by the following sequence: DNA to RNA to proteins, down to enzymes, down to catalyzed reactions, down to or across to hereditary characteristics. Again, DNA, RNA, proteins could go across to structural proteins, could go across to hereditary characteristics. Each of us is the sum of our genetic characteristics. Blood type, hair color, muscle protein, nerve cells, and thousands of other aspects of our structure and functioning have their basis in the genetic code of DNA. You may recall from the last chapter that day-to-day mistakes made in DNA structure are usually quickly corrected by enzymes. These mistakes might be likened to typographical errors made while typing a paper. If we are the enzymes, take a few moments to proofread, we can fix those typos. If we misspell a word, we can correct it. However, if our keyboard is missing a letter or two, or some keys don't work, then some words can never be spelled properly. In our cells, DNA is the code for protein synthesis. DNA is the keyword or the keyboard for the spelling of proteins. The sequence of amino acids. A mistake in DNA is incorrect bases or triplets of bases in the genes a person was born with, in parentheses, the keyboard, close parentheses, and cannot be fixed. This mistake will be copied by the mRNA. The result is incorrect or even missing amino acids, meaning the formation of a malfunctioning or non-malfunctioning protein. For an affected person, some proteins can never be spelled properly, or a particular aspect of cellular function is lost or impaired. This is called a genetic or hereditary disease, and a specific example is described in box 3.2, genetic disease, sickle cell anemia. The correction of these keyboard DNA mistakes is the focus of a great deal of research. The goal is to cure genetic diseases in individuals and perhaps even prevent, or at least limit, the further transmission of these diseases from one generation to the next. A simple yet exceptional molecular tool for this purpose is described in box 3.3. CRISPR/case 9 something borrowed. Takeaway: 1. State a physical difference between DNA and RNA. 2. State a chemical difference between DNA and RNA. 3. State a difference in the location of functioning DNA and RNA. 4. Besides being the site of protein synthesis, what other function do ribosomes have in this process? Although both types involve cell reproduction, their purposes are very different. Mitosis in the cell cycle Each of us began life as one cell, a fertilized egg. Each of us now consists of trillions of cells produced by the process of mitosis. In mitosis, one cell with the diploid number of chromosomes, the usual number 46 for humans, divides into two identical cells, each with the diploid number of chromosomes. This production of identical cells is necessary for growth or for the growth of the organism and for repair of tissues. Before mitosis can take place, a cell must have two complete set of chromosomes because each new cell must have the diploid number. The process of DNA replication enables each chromosome in the form of chromatin to make a copy of itself. This takes place during interphase, the time between mitotic divisions. Although interphase is sometimes referred to as the resting stage, resting means not dividing rather than inactive. The cell is quite actively producing a second set of chromosomes and storing energy in ATP. The long, thin, and invisible chromatin molecules then begin to coil very precisely and extensively. And if we were looking at the nucleus of a living cell under a microscope, we would see the duplicated chromosomes appear. Each would look somewhat like the letter X because the original DNA molecule and its copy, now called chromatids, are still attached at the region called the centromere. The stages of mitosis are prophase and metaphase and anaphase and telophase. What happens in each of these stages is described in Table 3.4. As you read the events of each stage, refer to Figure 3.5, which depicts mitosis in a cell with a diploid number of 4. Note, the events are listed in a table because they really are steps in the process. Your instructor will let you know how much detail of the various stages you will be responsible for learning. Not sure? Be sure to ask. As mentioned previously, mitosis is essential for repair of tissues because it replaces damaged or dead cells. Some examples may help illustrate this. In several areas of the body, mitosis takes place constantly. These sites include the epidermis of the skin, the stomach lining, and the red bone marrow. For each of these sites, there is a specific reason why this constant mitosis is necessary. What happens to the surface of the skin? The dead outer cell flake off or are worn off by contact with the environment. Mitosis of the epidermal cells in the lower living layer replaces these cells, and the epidermis maintains its normal thickness. The stomach lining, although internal, is also constantly worn away. Gastric juice, especially hydrochloric acid, is very damaging to cells. Rapid mitosis of the several kinds of lining cells replaces damaged cells and keep the stomach lining intact. One of the functions of red bone marrow is the production of red blood cells. Because red blood cells have a lifespan of only about 120 days, new ones are needed to replace the older ones. The older ones that die. Very rapid mitosis in the red bone marrow produces approximately 2 million new red blood cells every second. These dividing cells in the red bone marrow are among the stem cells present in the body. A stem cell is an unspecialized cell that may develop into several different kinds of cells. Stem cells in the red bone marrow may become red blood cells, white blood cells, or platelets. These marrow stem cells are often called adult stem cells, and many, if not all, of the body's organs have such cells. Embryonic stem cells will be described in a later chapter. These are cells in which all of the DNA still has the potential to be active. They may become any of the more than 200 different kinds of human cells. The stem cells found in the umbilical cords of newborns are between the adult and embryonic cells in terms of their potential. It is also important to be aware of the areas of the body where mitosis does not usually take place. In an adult, most muscle cells and neurons, nerve cells, do not reproduce themselves. If the cells die, their functions are also lost. Someone whose spinal cord has been severed will have paralysis and loss of sensation below the level of the injury. The spinal cord neurons do not undergo mitosis to replace the ones that were lost, and such an injury is permanent. The neurons of peripheral nerves, the spinal nerves, and cranial nerves to the head and extremities do not reproduce themselves. Neurons that have been damaged cannot be replaced. Skeletal muscle cells are capable of limited mitosis for repair. The heart is made of cardiac muscle cells, which, like many neurons, do not seem to be capable of mitosis. A heart attack, in parentheses, myocardial infarction, close parentheses, means that a portion of cardiac muscle dies because of lack of oxygen. These cells are not replaced, and the heart becomes a less effective pump. If a large enough area of the heart muscle dies, the heart attack is fatal. Research has found evidence for ongoing mitosis in the heart and the potential for mitosis after damage in both the central nervous system and the heart. Such cell division may be that of neurons or muscle cells that were stimulated to divide by chemicals from the adjacent damaged tissue, or the dividing cells may be stem cells that are among the specialized cells. A region of the brain that definitely has cells capable of division is the hippocampus, a structure that is found in both sides of the brain and that is necessary to form new memories and keep old ones. Other areas of the brain have been shown to be able to produce new neurons in particular circumstances. Regular and frequent exercise is one stimulus for the production of new brain neurons in mice. Mice are not people, but are mammals, as we are, and it is reasonable to try to determine with certainty if human brain cells, whether mature neurons or stem cells, also have this ability and will respond to the stimulus of exercise in a similar way. As for repair, at present we do not know that for most people with heart damage or central nervous system injury, mitosis does not take place rapidly, or at least not sufficiently fast enough to replace the cells that have died and preserve or restore normal functioning of the organ. Research is continuing and may eventually find the stimulus necessary to produce extended mitosis that will bring about true tissue repair. The cell cycle. The lifespan of a cell is specific and is genetically programmed. Some cells divide as part of their life cycle. Others do not. They simply carry out their functions at the end of chemical signals, which may be internal or come from other cells, cause a cell to produce enzymes that destroy its own nucleus. Again, at the end of a cell's lifespan, the death of a cell is programmed as well. Chemical signs, in parentheses, which may be internal or come from other cells, close parentheses, cause a cell to produce enzymes that destroy its own nucleus. The nucleus damaged cell begins to shrivel and is detected by phagocytic white blood cells, which clean up the cellular remains. This self-destruction or programmed cell death is called apoptosis and is essential for the normal functioning of tissues, organs, and ultimately the entire body. Some examples will be helpful. As you probably know, hair grows in tubular structures called follicles. A hair root in the follicle is active for a certain length of time and then becomes inactive. The cells at the root of the hair die by apoptosis. The old hair falls out and a new hair begins to grow. An example of greater importance is the development of the brain. The brain of an infant contains many extra neurons. The neurons that do not make connections with other nerve cells die by apoptosis and are removed. This process prunes the nerve pathways of the brain for the most efficient functioning. A third example, also of great importance, is the fetal immune system, which will have to distinguish non-self pathogens from the cells that are self, that is, part of the body. White blood cells, the lymphocytes, that do not learn to recognize the body's own cells must be destroyed, and they are triggered into apoptosis. One other aspect of apoptosis is significant. Although the process damages cells and attracts phagocytes, inflammation does not occur, as it would if those same cells were damaged by infection or physical trauma such as a burn. As mentioned earlier, inflammation is a beneficial process, but excessive inflammation can damage healthy surrounding tissue and stimulate unnecessary, and therefore even more damaging, immune functioning. No such damage occurs around cells that die of apoptosis. Meiosis. Meiosis is a more complex process of cell division that results in the formation of gametes, which are egg and sperm cells. In a cell about to undergo meiosis, DNA replication takes place, and then the cell divides into two cells. This is called meiosis 1. Each of these cells still have the diploid number of chromosomes, and each divides into two cells. This is called meiosis 2. DNA replication does not take place before meiosis 2, so each of our 4 cells will have half the usual number of chromosomes. Stated most simply, in meiosis, one cell with the diploid number of chromosomes divides twice to form 4 cells, each with the haploid number, half the usual number of chromosomes. In women, meiosis takes place in the ovaries and is called oogenesis. In men, meiosis takes place in the testes and is called spermatogenesis. This difference between oogenesis and spermatogenesis will be discussed and illustrated in Chapter 20, the reproductive systems. The egg and sperm cell produced by meiosis have the haploid number of chromosomes, which is 23 for humans. Meiosis is sometimes called reduction division because the division process reduces the chromosome number in the egg or sperm. Then, during fertilization, in which the egg nucleus unites with the sperm nucleus, 23 chromosomes of the sperm plus the 23 chromosomes of the egg will restore the diploid number of 46 in the fertilized egg. Thus, the proper chromosome number is maintained in the cells of the new individual. Takeaways. 1. What aspect of mitosis require ATP? Where does the ATP from mitosis come from? 3. In meiosis, one cell with the diploid number of chromosomes divides how many times? How many times does DNA replication take place? Why is this important? Aging and cells. Multicellular organisms, including people, age and eventually die. Our cells do not have infinite lifespans. It has been proposed that some cells capable of mitosis are limited to a certain number of divisions. That is, every division is sort of a tick-tock of a biological clock. We do not yet know exactly what this cellular biological countdown is. There is evidence that the ends of chromosomes, short sequences of DNA called telomeres, may be an aspect of it, which with each cell division, part of the telomeres is lost, in parentheses, rather like a piece of rope fraying at both ends, close parentheses. And eventually the telomeres are gone, or telomeres are gone. With the next division, the ends of the chromosomes' actual genes begin to be lost. This may be one signal that a cell's lifespan has come to an end. There are probably many different kinds of signals. Cellular aging also involves the inevitable deterioration of membranes and cell organelles, just as organelles. Just as the parts of a car break down in time, so too will cells. Unlike cars or machines, however, cells can often repair themselves, but they do have limits. As cells age, structural proteins break down and are not replaced, or necessary enzymes are not synthesized. Proteins called chaperones, which are responsible for the proper folding of many other proteins and for the repair of disposal and damaged proteins, no longer function as well as cells age. Without chaperones, damaged proteins accumulate within the cells and disrupt normal cellular processes. Clinical manifestation of impaired chaperones include cataracts and neurodegenerative diseases such as Alzheimer's disease, Parkinson's disease, and Huntington's disease. Much about the chemistry of the aging process remains a mystery, though we can describe what happens to organs and to the body as a whole. Each of the following chapters on the body systems provide a brief description of how aging affects the system. Keep in mind that a system is the sum of its cells and tissues and organs, and that aging is ultimately at the cellular level. Summary. As mentioned at the beginning of this chapter, human cells work closely together and function interdependently. Each type of human cells makes a contribution to the body as a whole. Usually, however, cells do not function as individuals, but rather in groups. Groups of cells with similar structure and function tissues, which is the next level of organization.